Hello people, how are you? In this video, we want to look at Trichomonas vaginalis. So as the name suggests, it is going to live in the vagina. This is a topic under parasitology. So it is a parasite, correct? So now this Trichomonas vaginalis, basically it is a protozoa. So it will be a single-celled organism, right? And it causes something called as Trichomonas vaginalis itself, trichomoniasis. That's what you can say. Trichomoniasis, okay? Basically, this is present a lot in these sex workers, okay? It can be transmitted from male to female and female to male. It lives in the vagina and the cervix of the female and urethra, right? So it can be in men and in women. Now, let's move on to the morphology of Trichomonas vaginalis. Look at the morphology. It occurs only in trophozoite form, guys, and it is a pear-shaped, you can see already in this diagram, it is pear-shaped. And it has 5 flagella and undulating membrane. Wait, let's confirm this. So it actually has 5 flagella, 4 normal flagella, free flagella, 4 free flagella and 1 undulating membrane. So look at this pear-shaped trophozoite. It occurs only in trophozoite form, no cyst, so that it doesn't want to trouble students. You learn only the trophozoite form, it's saying. So there are 4 flagella here. Guys, watch here. This is the free flagella. 4 free flagella are there. And this is the undulating membrane. Can you see the undulating membrane? Should we mark it again? This is the undulating membrane. Okay. So now what you have to understand is, this is a pear-shaped protozoa. It is having 4 flagella, 1 undulating membrane. This central one, whatever you're seeing here, this is the axostyle. What is this? Axostyle. It has just 1 nucleus, and whatever you're seeing here, these are food vacuoles. Here again, we'll zoom this. Look at this, food vacuole with bacteria. So this is the morphology. Okay. This axostyle is there, right? It projects downward like a tail. Can you see here? It projects downwards like a tail. And this undulating membrane is there, right? That is supported at the base by something called as a costa. You see the costa here? This is the costa. Costa. It is supporting the undulating membrane. So let's write these points, guys. Guys, we have to write these points, otherwise you will not remember. So basically, the undulating membrane at base has support called costa. Okay. And the axostyle projects downwards, projects posteriorly like a tail. Okay. Now coming to its motility, the motility is rapid, jerky or twitching type. Guys, rapid, jerky or twitching type of movement it has. If you want, you can remember that, okay? That completes the morphology of Trichomonas vaginalis. Are you paying attention? What are we looking at today? Trichomonas vaginalis. Very good. So now let's move on. The next thing we will look at is the life cycle. The life cycle of Trichomonas vaginalis is completed in a single host, either in male or female. So it can either live in male or female, and it has single host to complete life cycle. And when we are saying male or female, it is human, male or female. Single host is enough to complete life cycle, whether male or female. Okay, then mode of transmission. The trophozoite cannot survive outside, so infection has to be transmitted directly from person to person. Sexual transmission is the usual mode of infection. So it is sexually transmitted. So these people can have other sexually transmitted diseases. So it can coexist with other sexually transmitted diseases like candidiasis, gonorrhea, syphilis, HIV, etc. Okay. Then even during birth, a baby, a neonate may get infected. Then fomites such as towels, handkerchiefs have been implicated for transmission. So it's just not sexually transmitted. It can be transmitted via fomites also. Fomites like towel, handkerchief, etc. So there's only trophozoite form, you know, and how they multiply. They multiply with binary fission, guys. They multiply with binary fission. They multiply with binary fission. As cysts are not formed, trophozoite itself is the infective form. So what and all you should know, trophozoite is infective form. No cysts are there. No cysts are there. Okay, then no cysts. Cysts are not formed. Okay, then they divide by binary fission. Okay, incubation period is 10 days. Incubation period is 10 days. So if a person is infected, then you can guess that 10 days back he had some intercourse probably with an infected person. Okay, then the life cycle of Trichomonas vaginalis is done. Now we are moving on to the pathogenesis. Okay, so let's move on to the pathogenesis. Let's move on to the pathogenesis of this Trichomonas vaginalis. So basically the pathogenesis is something like this. It infects squamous epithelium and not columnar epithelium. So where is the squamous epithelium present? So it secretes cystine, proteases, lactic acid, acetic acid. Okay, so it is going to secrete all these kind of enzymes, and it disrupts the glycogen levels. So it infects the squamous epithelium, right? So it likes squamous epithelium. It secretes all these enzymes, cystine, proteases, lactic acid, acetic acid, and it disrupts the glycogen levels, right? It disrupts the glycogen levels, and this always lives in the host, so never outside. So it has to go from the male to female, female to male, or whoever is having intercourse. So it is an obligate parasite, just like virus, right? Even viruses they cannot live outside the host. These cause petechial hemorrhages like strawberry mucosa. See, there's a photo here of the strawberry mucosa of the cervix. So they cause petechial hemorrhages, metaplastic changes, desquamation of the vaginal epithelium. So you know what metaplastic changes are, right? Like squamous can become columnar or something like that. So they cause metaplastic changes, desquamation of vaginal epithelium. So they cause intracellular edema. They called as chicken-like epithelium. Some strange words are there here, like strawberry mucosa, chicken-like epithelium. What and all the Trichomonas vaginalis will do. So we're done with the pathogenesis. We're moving on to the clinical features of Trichomonas vaginalis. Hello people, now let us look at the clinical features of Trichomonas vaginalis. In men, they are very lucky. Sometimes it's just asymptomatic in men. Otherwise, it can cause urethritis, epididymitis, and prostatitis. So inflammation of urethra, epididymis, prostate, everything. Okay, so that's in men. In females, what will happen? It will affect the vagina. It will affect the vagina. It will affect the cervix. It will affect the uterus, endometritis. It can affect neonates. Okay, that is neonates means just born babies. So they can also develop issues. Let us look at the issues in females first. So it leads to pruritic vaginitis, itchy vaginitis with offensive greenish yellow discharge. Remember, greenish yellow discharge. Let us go here and find a nice greenish yellow. Green is here, yellow is here, greenish yellow is here. What do you say? We pick up this color. A greenish yellow discharge. Unfortunately, you can't see it. Greenish yellow discharge. Often frothy discharge. There will be dysuria, dyspareunia. What is that? As expected, it is painful sexual intercourse. Painful sexual intercourse is called as dyspareunia. Periunia. Okay, what happens to the cervix? You already saw it causes strawberry mucosa and cervical erosion. Endometritis is kind of infrequent. What is PyoSalpingitis? You know what salpingitis is, right? Pyo is actually pus, okay, and salpingitis is something to do with the fallopian tube. So there will be painful suppurative inflammation of the fallopian tube, okay, but these are very infrequent. They're very rare complications, okay. Then coming to the neonates who are born to the infected mother, they can have pneumonia and conjunctivitis. So here you can see something very, very interesting, that it is not actually infecting the vagina or the genitals. So it can also infect the lungs and the eyes. Incubation period, we already saw 10 days. In one paragraph in this textbook, they have given one way. I don't know what to do. So we're just sticking to the old one, 10 days, plus or minus 6 days or whatever. Now coming to the diagnosis of Trichomonas vaginalis. How will you diagnose? People, so now let's move on to the lab diagnosis. Is that fine? So just take a deep breath. Okay, now let us look at the lab diagnosis of Trichomonas vaginalis. So basically microscopic examination. So what you will do, you will take the vaginal or the urethral discharge and examine it microscopically in saline wet mount. Okay. In saline wet mount preparation, you will look for the characteristic jerky and twitching motility, and the shape also you will look for. In males, the trophozoites may be found in urine and prostatic secretions also. So what at all you can collect? You can collect vaginal or urethral discharge, urine, prostatic secretions, male urine, prostatic secretions. So summarizing here, the specimens you can collect are the vaginal discharge, urethral discharge, urine from male you can take the prostatic secretions. Microscopic examination by saline wet mount microscope, where you will look for the characteristic jerky and twitching motility. In fixed smear also you can look for these trophozoites. You can stain with acridine orange. Let's put an orange here. Acridine orange, please. Orange. Acridine orange. Can't see this orange. Okay, let's go with this. Some orange. Papanicolau and Giemsa stain. So you can use all these stains. Then you can also do direct fluorescent antibody. Direct fluorescent antibody is another method of detection of parasite, and it is more sensitive. Okay. This is more sensitive than wet mount. Now moving on to the culture. The culture is the gold standard. It is recommended when direct microscopy is the negative. So you will do it if you think direct microscopy you didn't find anything, then you do some culture. Okay. It is the gold standard as well as the most sensitive, 95% sensitive method for the diagnosis of Trichomonas vaginalis infection. So Trichomonas vaginalis, it grows best at 35 degrees centigrade to 37 degrees centigrade and anaerobic. It grows in anaerobic conditions. So just pay attention here. It grows in anaerobic conditions. Trichomonas vaginalis. Okay. Trichomonas vaginalis, TV, grows in anaerobic conditions. So you should know that the optimal pH is like 5.5 to 6.0. So we saw the optimal pH of vagina. It's around 4, right? Didn't we? Okay. Then moving on, so we are looking at the culture here. It grows in anaerobic conditions. It can be grown in a variety of solid or liquid media. It can be grown in tissue culture, eggs. Cystine, peptone, liver maltose, CPLM medium and plastic envelope medium, PEM, are often used. So many types of media are used, so many words they told here. So this is culture is the gold standard. It is most sensitive, right? 95%. It grows at what temperature? Room temperature? No, it grows at body temperature, 35 to 37 degrees centigrade. The optimal pH they are saying is around 5 to 6. I don't understand this because the vagina pH is, they have told is around 4. And it can be grown on so many types of solid and liquid media, tissue culture, eggs. So just remember CPLM media. CPLM media is cystine, peptone, liver maltose medium. So are you able to see this or it's too small for you? Let me just zoom in. So it can grow in cystine, peptone, liver, maltose medium. Okay, and it can also grow in PEM medium, that is plastic envelope medium. Okay, so they are using all these for culture. So we are done with culture. Let's move on now. Serology, guys. Serology, you will use ELISA to demonstrate the antigen of Trichomonas vaginalis. In the vaginal smear, you can demonstrate the antigen using monoclonal antibody for 65 kDa surface polypeptide of Trichomonas vaginalis. So if you want to know the details, you can use monoclonal antibody for 65 kDa surface polypeptide of Trichomonas vaginalis. Okay. So you can demonstrate the antigen of demonstrate the antigen in vaginal smear by ELISA by using monoclonal antibody for 65 kDa surface polypeptide. So this is the antigen of Trichomonas vaginalis. Okay, so this is the antigen. Which one? 65 kDa surface polypeptide. Okay. Moving on to molecular methods. In molecular methods, definitely you will go to DNA level and do everything. So in molecular method, how will you detect? DNA hybridization and PCR are highly sensitive and specific. These are obviously highly specific and sensitive. Why? Because they are going at the DNA level. So obviously it is going to be good for detection and diagnosis of trichomoniasis. So what and all did they say here? DNA hybridization and PCR. DNA hybridization and PCR are highly sensitive. Okay. Obviously they are highly sensitive. So now we are done with the lab diagnosis of Trichomonas vaginalis. Happy? Are you happy? Did you enjoy the lab diagnosis? Now shall we move on to the treatment? Take a break. Okay. So now let us go on to the Trichomonas vaginalis treatment. The treatment is actually Metronidazole. Okay. It is the drug of choice and it can be given to both the partners. You don't give it only to one person, give it to the other partner also. Metronidazole, single dose orally, okay, or you can give 500 mg twice orally a day for 7 days is the drug of choice. So you can give 2 gram single dose, 2 gram single dose or 500 milligram orally twice a day for 7 days. Okay, this is the drug of choice, metronidazole. If patient is not responding to this treatment, then you can increase the dose of metronidazole or you can even give it parenterally. In pregnancy, metronidazole is safe in 2nd and 3rd trimesters only, not in the 1st trimesters. Okay, so don't give metronidazole in 1st trimester. Don't give in 1st trimester. Okay, how do you prevent? So let us put a word here on prevention or prophylaxis, that is prevention of Trichomonas vaginalis infection. So basically you can use condom, etc., some barriers to make sure that just like other sexually transmitted diseases, avoid sexual contact with infected people and use the barrier whenever there is intercourse. You should also test the sexual partner for Trichomonas vaginalis. Okay, that's all. So we are done with Trichomonas vaginalis. Let's take a quick recap. Trichomonas vaginalis, we are looking at it at the perspective of parasitology. It is a protozoa. It causes trichomoniasis. The habitat is the vagina and cervix of female and the urethra. 75% sex workers are known to have this Trichomonas vaginalis infection. Male can pass to female, female can pass to male. Trichomonas, it usually occurs only in trophozoite form. That is to make sure that exam for us it will become easy. It is a pear-shaped protozoan. Just thank Trichomonas vaginalis. Take a second and say Trichomonas vaginalis. I'm so thankful that you have only trophozoite form. I have to draw only this diagram in the exam. It has 5 flagella, 4 free and 1 undulating membrane which is supported at the base by costa. There is then this axostyle which has projects like a tail in the posterior. There are siderophoric granules. There are food vacuoles which contain bacteria. There is just 1 nucleus. The motility is rapid, jerky or twitching type. The life cycle of Trichomonas vaginalis, from male to female it can go. It can complete its life cycle either in male or in female. Single host is enough for it to complete its life cycle. It is a sexually transmitted disease. It can also be transmitted from fomites like towels. Even during birth, a neonate can get the infection from its mother if the mother was infected. So usually this condition coexists with other STDs. So definitely you'll have to, if a person has Trichomonas vaginalis, you can check them for other STDs. The trophozoite itself is the infective form, obviously because there are no cyst forms, right? They divide by binary fission. Moving on. It loves squamous epithelium. It secretes a lot of enzymes like cysteine proteases. What was the cysteine? Is the spelling correct? Cysteine proteases. Pathogenesis. Yes, pathogenesis. Cysteine proteases, lactic acid, acetic acid and all it secretes. It disrupts the glycogen level. It increases the pH of vaginal fluid. Guys, it's secreting all these acids. It is increasing the pH of the vaginal fluid. Okay. So it likes the pH if it is above this pH, right, 4.5, then only it likes. So it will be like around 5, 6 only it likes. That's we have seen, right? So we have to correct this, guys. It increases the pH of the vaginal fluid, okay? It causes the strawberry mucosa, petechial hemorrhage of the cervix. Metaplastic changes can be there in the cervix. Then intracellular edema, chicken-like epithelium can be there. Clinical features often asymptomatic in males. It causes urethritis, epididymitis, prostatitis. In females, it will lead to vaginitis. Offensive greenish yellow discharge can be there. Frothy discharge. Painful sexual intercourse. Erosion of the cervix. Strawberry mucosa. Endometritis. Pneumonia, conjunctivitis in neonate. Now lab diagnosis, how will we do? You will collect the specimen, then microscopic examination and saline wet mount. You will observe the characteristic twitching motility. Fixed smear. See this, 1 second, hold on. So if you are using fixed smear, you can use stains like Acridine Orange, Jimsa stain, etc. Direct fluorescent antibody DFA is more sensitive than wet mount, they are saying. Gold standard is the culture actually. So you will give it anaerobic condition, very important, you should give it anaerobic conditions. Then the media you will use will be CPLM media, that is cysteine peptone liver maltose medium, and PEM, that is plastic envelope medium. Okay. So these are the media you will use for Trichomonas vaginalis. Serology, you can demonstrate the antigen, that is 65 kDa surface polypeptide of Trichomonas vaginalis by ELISA. Molecular method standard, that is DNA hybridization and PCR. Okay. Treatment of drug of choice is metronidazole, guys. Don't give it in first trimester. Prophylaxis barrier method, safe sex. Okay. That's all for now, guys. Hope you enjoyed Trichomonas vaginalis. So this is a perfect bye from Trichomonas vaginalis. Bye bye.